Okay, folks, welcome back. And this is the course on bioethical issues at the beginning of human life within the Masters of Science in Bioethics at St. Thomas University. And today is our last lecture on this series, on this course. Okay, as I explained briefly previously, next Saturday, we don't have class as such because we're in the transition for the other course, bioethical issues at the end of human life. So because that Saturday morning is open, next Saturday morning, you're very welcome to come to the forest and do some work there, restoration of the original environment. We might be working in the Chicky Hut actually, which is next to the School of Business, and there's plenty of parking there, especially on a Saturday morning. And might as well finish this announcement. I'll be there starting around 9 a.m., okay, for the duration of the day. Well, probably until maybe 1 or 2 p.m. So you can come anytime. And remember that this will count as part of the internship, 3 for 1, 3 hours of internship for 1 hour of labor. So you come equipped accordingly. We'll be outside. If you have a white rim hat that will cover your ears and the neck, you can also get sunblock because we'll be in and out of the sun back and forth. Long sleeve shirt and long pants, socks, so you don't get any stickies with weeds and stuff like that. And then also shoes, either tennis shoes or boots or something like that. No flip-flops and no high heels out there. Okay, so just come equipped for outdoors. If you have some gardening gloves, you can bring that. Otherwise, I'll have a pair for you to use that day. So this is next Saturday, beginning at 9 a.m. You can just gather at the first parking lot of the School of Business as you drive in. It would be like the west parking lot of the business building. Okay, thank you. So this last lecture, it's always kind of at the forefront of what's happening today and other issues of manipulation of human embryos, issues at the beginning of human life, right? And so it's kind of a panoply of themes, techniques that are come up over the horizon. I say not a week goes by without some kind of bioethical issue arising, either at the beginning of life, at the end of human life, in healthcare or environmental bioethics, right? Just last week I saw a news in the BBC. I usually look at the BBC, just the headlines in the morning, early in the morning, see what's happening. And there was an article there of selling plots of the Amazon forest on Facebook. Just like that. It's a scheme, it's a scam. Just like a century ago, plots in the Everglades were being sold to people up north. There was no online back then. You know, those were actual titles and deeds that they got for plots in the Everglades. And then when those people came down south, when Flagler built a railroad all the way down here in the 20s and 30s, and they went to look for their lot, right, their precious lot, their dream lot, paradise, tropical paradise and all this, it was in the middle of the Everglades. It was the swamp. So it was a big scheme, big scandal that went on a century ago. And today we have basically the same thing, selling away the Amazon forest online, right? Facebook, they'll give you a nice little map of your plot from a satellite image, an actual photograph of the thing. Okay, so the issues never stop. Anyway, today we're going to cover several of these which are heavily influenced by technology. Again, as it stands to reason because as technology continues to advance, well, creativity of the mind, right? And what used to be science fiction a few years ago is now becoming a reality in the lab or maybe even in the field. So we're going to look at several techniques, one that is called 3-parent embryos, another one, chimeras, between human and non-human chimeras, animals, prenatal diagnosis, which is already pretty standard already in medicine, prenatal diagnosis, not pre-implantation diagnosis, but prenatal, in other words, with the pregnancy already happening. And then growing human organs in animal models is another possibility. And finally, to end up with kind of a positive note, we'll look at fetal medicine and fetal surgery, maternal fetal medicine and surgery that is going on right now, okay, for actually healing in the womb. Okay, so moving forward, as always, it's a two-step thing. First, we have to do the science, the biology of it, and then the bioethical analysis based on that. So we're going to look at 3-parent embryos first. And this is mostly because of a cluster of diseases that is called mitochondrial diseases. Okay, mitochondrial diseases. You can look it up later online, but basically the mitochondria is the energy factory of the cell. We'll review a little bit this biology in a minute, but just to keep in mind that these are organelles that are inside the cell, and we all have them. We have hundreds of thousands of them in each cell, and they are the energy factory of the cell. Biochemically, they recharge ATPs, and ATP, adenosine triphosphate, is the energy molecule of the cell, all right? It is equivalent to a little battery that provides energy for all the metabolic processes that are going on at the cellular level. So let me back out for a moment and look a little bit at the hierarchy of life for the benefit of those. So as always, on the biological side, some of you have a greater, larger biological background, even degrees in it. So this is kind of review. Others don't have that much of a background in biology, maybe high school biology a few years ago. And so this is a general review. Okay, so bear with me. There's what we call the hierarchy of life, starting with the cell. The cell is the basic unit of life. And this is also a good review for the summer as we go look at the environmental bioethics course, because we'll get more into nature in that course. But basically the cell, the living cell is the basic unit of life, all right? But it turns out that most cells are not living on their own, just sitting on top of a counter there, unless they are bacteria or protozoans, which is that grab bag of various microorganisms like the amoeba, the paramecium, the volvox, etc. And even many of those form little colonies. But basically cells, especially in what we call metazoans, the ones who are more than a single cell, individuals that are more than single cell, and that includes the animals, the plants, and the fungi, the mushrooms as a third kingdom, okay? We're all made up of cells, and the basic unit of life, what is alive, is the cell. Now, when cells come together, they form tissues. Cells that have a similar function form tissues. For example, skin cells, they form the skin, right? Then tissues that have a similar function, they come together to form organs. Like, for example, a liver tissue will form the organ of the liver itself. And then organs coming together, organs that have a similar function, they form what we call organ systems, but systems. So we can look, for example, at the digestive system. The digestive system is made up with a number of organs, and we can follow them sequentially, right? For example, the mouth, to start with all the chewing mechanism and the saliva that is secreted to make the bolus. The bolus is the material, the chewed material food that is going to be digested. So that's the first organ, say, in the digestive system, then goes down the esophagus, another organ, and then the stomach, and then the intestines and the colon. So you see that these are different organs, but they're all involved in digestion. and they form the digestive system, right? So we have about a dozen digestive systems in the body, depending on how they're classified structurally or functionally. So structurally, we can talk about the muscular system and the skeletal system as two systems. But functionally, many, especially in the medical field, they talk about the skeletal muscular system or the musculoskeletal system in coordination, because they're both involved in movement. You see, so anyway, from half a dozen to a dozen organ systems in the human body, and that forms the whole organism. What is interesting about the organism as a whole is that those systems have separate different functions. So when we get to the level of system and jumping to the organism, who is the actual living creature, right? Then their systems have different function, separate function. And so the organism actually integrates all those various systems into a living whole. And typically there is a coordinating system, a coordinating system, the one that coordinates all the other body functions, all the other systems. And in animals, that is the nervous system. Okay, the nervous system is the one that coordinates the functioning of all the other systems in unison, in synchrony, so they will have a living organism. Plants lack a nervous system, and so they have, they are more primitive that way and prior to the animals. They also have what is known as undeterminate growth, as opposed to animals that have determinate growth. In other words, our arms come out on the top part of the trunk and our legs come out of the bottom part of the trunk, whereas in branches on a tree, well, you can cut one branch off and a tree will put out a branch somewhere else. So that's indeterminate growth. At any rate, back to the cell which is the basic unit of life. Just like we have organs, like an organism has organs, the cell also has organelles, okay? Organelles that are microscopic, of course, because they're inside a cell that is already microscopic. But these organelles are structures that have particular functions. So just like different organs of the body have different functions, the organelles also have different functions, okay? The most prominent organelle in a cell typically is going to be what? The one that holds the DNA, the nucleus, all right? And the nucleus is crucial because it's like a safety box. It's like a safe, right? It maintains the DNA, the genetic material, safe in there. And in fact, it has a double membrane, just like the cell membrane is also a double membrane. So there is speculation that it may have started also by the endobiotic theory that we saw last semester, endobiosis. But then there are other organelles that also have double membrane. The mitochondria is one of them. You can see that it has an external membrane that is rather smooth, but then the internal membrane is corrugated, is folded. It has these folds that are called cristae. And that is, what is your intuition when you have a membrane that has a lot of convolutions, a lot of folding? What is the function? To increase what? Surface, exactly, just like the brain, the convolutions of the cortex, which when we expand the brain takes up a much larger surface, okay, because that's where the thinking process actually occurs, and the analysis of stimuli and interpreting them into something intelligible. And so the increase in surface with corrugation within a constricted space, all right? So here's a diagram of the cell. Here is a single mitochondrion, again from the Latin, one mitochondrion, several mitochondria. Here's an actual photograph of the cell of a mitochondrion. This is a micrograph, an electron micrograph, cross section. On the outside here, actually, these little dots are ribosomes. There are other structures, organelles. The ribosomes are crucial for making what? They make proteins, all right? So they interpreted the mRNA, just like in the vaccine now with the COVID vaccine. That mRNA goes to the ribosomes, and the ribosomes reads out that mRNA, and that messenger RNA codes for polypeptides that make up proteins. So every part of the cell, every compartment has its own function, and it's all held together by a skeleton, microtubules and fibers that are microscopic, obviously almost down to the molecular level here, and typically translucent or transparent, but they're there. So they give kind of a scaffolding to the cell so that the cells are not just, the organelles are not just wiggling around inside according to where gravity is or the motion of the individual, all right? There is an internal skeleton that is a soft gelatinous skeleton that keeps the organelles in place. So you can think of a cell, cytoplasm, something like jello or jelly, where you put in marbles or whatever structures and they stay in place because of the jelly, the fibrous structure there itself. All right, so back to the mitochondrion. The mitochondrion is, again, the energy factory of the cell. This is where biochemically glucose is burned, and the release of energy from burning the glucose recharges ATPs, recharges adenosine triphosphates, and then the triphosphates go all over the cell to provide energy for protein synthesis and for all the metabolic processes, the cascades that occur in metabolic processes. So metabolism is just a fancy word for being alive, all right? And metabolism itself, you may know, is subdivided into two parts biochemically, either building molecules or breaking down molecules. So the building molecules is called anabolism and the breaking down is catabolism. Those are details, but basically metabolism is body functioning, cellular functioning, and therefore is what's keeping us alive. So biochemically life, organic life on Earth can be simplified, if you will, to chemical reactions. A humongous amount of chemical reactions that are occurring in an orderly fashion, okay? When those chemical reactions no longer occur in an orderly fashion, we have chaos, we have decay, and we have death. And so life is truly a humongous amount of chemical reactions that are occurring in every cell, every second of our life, without us really being aware or having any say in it, all right, in a sequential process, and this keeps us alive. And what is coordinating all that at the cellular level is the DNA. So the DNA and the nucleus is equivalent, it's analogous to the brain and the nervous system of the whole organism, all right? Okay, so back to the mitochondrion. Mitochondrion is another one of these other organelles that may have come about through the endosymbiotic process, again, because it has a double membrane, pretty much like the double membrane of the cell itself. And then the other one that is in plants, in addition to mitochondria, what is in plants also that is involved in photosynthesis? The chloroplast, okay? And they have, it's a reference, chloro is a reference to color. Plast, the plastid is, or a plasmid is it's a structure, it's an organelle. So the chloroplasts are in plant cells in addition to the other, but plants also have mitochondria because they're also living, they also respire, okay? And cellular respiration is the biochemical process that occurs here. Cellular respiration is just a fancy wording for burning glucose. And when we burn glucose, we get the energy release, which is what charges the ATPs, okay? Measured in kilocalories, etc., or calories. All right, now, mitochondria are in the cytoplasm and not in the nucleus of the cell. So there are basically two options for organelles. They're either in the nucleus or in the cytoplasm. And except for the DNA and the nucleolus, everything else is out in the cytoplasm, including the mitochondria. So because they're in the cytoplasm, at fertilization, all right, where do we find the bulk of the mitochondria? In the sperm cell or in the egg cell? In other words, another way of rephrasing this, which of those two gametes has more, much more cytoplasm? The sperm or the egg? The egg. The egg has much more cytoplasm by volume, all right, because the egg is intact, whole, including the nucleus, but the sperm is mostly just a nuclear capsule with a little bit of cytoplasm, and then the nuclear, and then the cell membrane, the flagellum, and all that. And it does have mitochondria. Actually, the sperm does have mitochondria. They're concentrated toward the base of the tail of the flagellum, because that's where most of the activity is, the motility. And that motility is burning glucose, is burning, using up ATPs. And so most of the mitochondria are concentrated at the base of the flagellum there. So the sperm does have some mitochondria, but not a lot. And when the sperm is emptied into the egg in the fertilization process, those few mitochondria do get emptied into the egg, all right, into the ovum. So there are some paternal mitochondria in the egg, in the fertilized egg, in the zygote, but it's a double whammy because not only does the sperm have very few mitochondria compared to the ovum, but also the ovum cytoplasm attacks the sperm mitochondria and digests it and attacks it as a foreign object. Somehow the nucleus of the sperm is able to evade that attack, right? But you consider for an egg, a sperm is a foreign object, right? That is penetrating it, and therefore needs to be attacked with digestive enzymes, proteases, and so forth. So the paternal mitochondria are attacked by the egg, and most of them are killed. A few survive, but the vast majority of our mitochondria are maternally inherited, you see, because the zygote will have mostly maternal mitochondria. I remember many years ago, decades ago, reading a paper actually in the New England Journal of Medicine, where some researchers had found paternal mitochondria in a zygote, all right, and in embryos, in human embryos. So that was evidence that some of those mitochondria do get to survive, but very, very few. The bulk of them are maternal. And so we have the bulk of the mitochondria, because every single cell of our body comes from the zygote, right? And the zygote is a fertilized egg. So the cytoplasm of the zygote is mostly maternal cytoplasm. Okay, which is where the mitochondria is. So the bottom line about all this is that our mitochondria are maternally inherited. And not just women, but men also, because we were all zygotes at one point. All right, so mitochondria are maternally inherited. What happens? Mitochondria also has its own DNA. Well, we saw that when we talked about mitochondrial Eve, right? Where we can trace the inheritance of our mitochondria back. We can create actually a phylogenetic lineage of all humanity just by looking at the mitochondrial DNA. And yes, that mitochondrial DNA codes mostly for the proteins and the factors that are needed in the cellular respiration process. Okay, so they are the DNA that is in there, the genes that are in the mitochondria are typically specific to the process, to the function of the mitochondria, which is recharging ATPs. Okay, so they're very specific. Now, mitochondria are maternally inherited, and they have DNA. And if they have DNA, any DNA can have mutations, all right? And so when that DNA gets mutations, then it's going to affect the function of the mitochondria. It can either affect the function in several ways. It can produce fewer mitochondria in the cells, right? So it would be a deficiency, just like there would be a insufficiency of sperm in some men that make them incompetent, incapable of fertilizing. So the same with mitochondria, analogously. If there are fewer mitochondria than the several thousand that we need, then the process of recharging ATPs is going to be weak, all right? And what is that? What is going to be the end result of functionally of that person as a whole when they experience some of these mitochondrial diseases? They're going to be tired. They're going to be tired. They don't have enough energy. So it could be a number of things. It could be that insufficient mitochondria are made, or it could be that the mitochondria themselves are not functioning 100%, you know, they're maybe at 50% because some factors are missing or malformed, etc., the conformation of the proteins. So it's a host, and that's why it's called mitochondrial diseases, right? It's a host of diseases, but the end result is that these people typically are tired, okay? It's not necessarily fatal, but it is debilitating and it becomes chronic, lifelong. So, because IVF already exists for decades, now some scientists, and leave it to the UK, in the UK, have come up a few years ago with this invention of 3-parent embryos to get rid of the mitochondrial disease. In other words, to have women who have the mitochondrial disease being able to bear children who don't have the disease, all right, who are normal, at least for that issue. So let's look at how that happens. Of course, it involves IVF because there's manipulation either of the egg or the zygote. And there are two options, depending on whether we manipulate the egg or the zygote. So one method is to repair the embryo. The other method is to repair the egg. So let's follow it through. Let's look at the embryo first. There is a woman whose eggs, well, they have to undergo in vitro because the egg or the zygote needs to be in the laboratory for manipulation, all right? So a woman undergoes hyperovulation. Now we have her eggs in the lab. Then a man donates a sperm, can be the husband or not the husband, catalog, etc., donor, anonymous. But now we have fertilized eggs, so we have a zygote. The zygote, of course, comes from a woman who has the mitochondrial disease. The father doesn't have the disease, all right? So this zygote carries the disease and will express because it's in the cytoplasm. Okay? The mitochondria of the zygote are diseased. They have the mitochondrial disease. So we bracket that for a moment. It's in the incubator. Now, this same man fertilizes the eggs from another woman who doesn't have the disease. This woman is an egg donor. This woman is an egg donor. So she undergoes also hyperovulation, and her eggs, which don't have the disease, her cytoplasm, right, does have the disease, is fertilized by the sperm of the same man. So it's the same father. All right. Then what happens? Essentially, a nuclear swap. A nuclear swap. In other words, from the mother-to-be, okay, the egg is, what is discarded here essentially is the cytoplasm. So the nucleus, the diploid nucleus of the zygote, the nucleus is good because the disease is in the cytoplasm, it's not in the nucleus. So the nucleus is, let's say, healthy, all right? Let me back up for a moment. First, we have to create a suitable place for the nucleus to go. It's a type of cloning, it's a nuclear swap. First, for this donated egg that was fertilized by the man, everything is normal here, but we don't want the donor woman nucleus. We want the mother-to-be nucleus. So this zygote is enucleated. This zygote here is enucleated. What do we have? Now we just have the shell. We have a cytoplasm that is normal because these mitochondria are normal, okay? So just follow, very straightforward. The donor zygote is enucleated, okay? And now we have a cytoplasm that is normal, normal mitochondria. And we take the nucleus from this couple, who are going to be the parents, and that nucleus goes into the cytoplasm of the donor, okay? That's the embryo swap. It's an embryo swap because and it's interesting that they call it embryo swap because they have to recognize that once we have a zygote, we have an embryo. Okay? In other words, once we have fertilization, we have an embryo, who is a zygote, which is a single cell. All right, so you see what happens here. We're using the cytoplasm of the donor woman, and we're using the nucleus of the couple. And what is discarded is the cytoplasm of the deceased mother, the mother who carries the mitochondrial disease. All right, so that cytoplasm is discarded. We end up with a reconstructed zygote here, nucleus from the father-mother couple, the genetic couple, and cytoplasm from the donor woman who donated just the eggs, the ova. And then that embryo, that zygote, goes into the incubator, 37 degrees centigrade for 3 to 5 days, blastocyst, attempted implantation into this woman who has the disease, but now the embryo doesn't have the disease. Okay, and that embryo, the embryo will then go through normal gestation and be born with normal mitochondria. Okay. Also because what protects the embryo from receiving mitochondria from the mother? The placenta. The placenta acts as a filter, okay? And nutrients will come from the placenta. Some cells do interchange, and there is now evidence that the fetus provides some cells to the mother body, and the mother's body provides some cells to the fetus also, so there's some interaction there, but they're going to be very few in comparison to the rest of the cells of the fetus. So the fact that a few cells in the fetal body that come from the mother are weak, but the other trillions of cells are normal, so that weakness is negligible. You follow? All right, so that's the biology of that. The alternative is to do the egg repair. In other words, before fertilization. At the end of the day, what are we looking for? In mitochondrial disease, what we need is a healthy cytoplasm, a healthy ovum at the cytoplasm level. So why don't we do the nuclear swap before fertilization? In other words, here's the mother-to-be, hyperovulation. We have her ova, which have the diseased mitochondria. Donor woman, hyperovulation, we have her ova, which are all normal, cytoplasm and nucleus. Then we enucleate the donor ova. Now we get the healthy cytoplasm, okay, but there's no fertilization. There's no fertilization. So now we get the shell, the healthy, clean shell of enucleated ova. And we do essentially a nuclear swap, but before the zygote. So we take the nucleus, the haploid nucleus of the mother-to-be, just the nucleus, which is healthy, and insert it into that cytoplasmic shell, right? The enucleated egg of the donor before fertilization. And then what do we discard? We discard the cytoplasm of the deceased mother, which is deceased mitochondria. We discard also the nucleus of the donor woman because we're not interested in her DNA. And then once we have that new reconstructed egg, new reconstructed ovum, with the donor cytoplasm and the mother-to-be nucleus, we fertilize that. All right, so there's only one fertilization. Now we have the zygote, incubator, 3 to 5 days, blastocyst, attempted implantation, etc. And the attempted implantation would be then theoretically on the cytoplasmic disease mother, but the embryo, again, is healthy because the cytoplasm of that embryo comes from the donor woman. Okay, so those are the two possible scenarios biologically. Bioethically, go back to scenario 1 here, all kinds of issues. First of all, in vitro. So already an intrinsic injustice. What happens, so 2 fertilizations. So that's essentially, you know, adultery, that's fertilization adultery. Because the same man is fertilizing 2 different women. All right, now, in the donor zygote, when we enucleate a zygote, we're taking out the nucleus, we're essentially killing that zygote, you know. Again, if the nucleus is analogous to the brain, it's like decerebrating someone, you know. So that kills that embryo, okay? There's a manipulation here. Of course, this is not going to be a single attempt, right? This is somatic cell nuclear transfer, or a type of nuclear transfer. And therefore, the average trials for Dolly was about 270, or I think it was 258. Anyway, it's in the 200s, because when they clone the cat, when they clone the dog, when they clone the sheep and other animals, it typically takes an average of about 200 trials to get one to actually implant and survive, et cetera, et cetera. So that means, you know, and we as mammals, we're no different. So we are talking about the possibility of 200 human beings killed to get this one healthy one in the process. Absolutely insane, all right? But it's a trial and error industry, people can pay for it, and it's utilitarian mentality. On the other side, on the second alternative, let's say that hypothetically enucleating an egg is not killing because there's no embryo yet, there's no fertilization, all right? It does involve in vitro, so it involves all the evils of in vitro. But then again, it's not going to be a single trial. It's going to be a series of trials to see which one actually plants and takes and makes it to birth. But the added negativity in all this is that we can ask the question, this embryo, this child, who's the father? There's one father. There's one father because the sperm per nucleus comes from one man, okay? But who's the mother? These embryos necessarily have a nuclear mother and a cytoplasmic mother, or 2 different individuals. Who's the mother? Okay, and both the nucleus and the cytoplasm are essential in the zygote for further development, so they're both mothers. They're both biological mothers, and that's why this is a 3-parent embryo. You know, and then are they going to tell the kid or not? Most people don't tell them, don't tell the children, and children don't know. But this child has 2 mothers for life. Okay, and if at some point the child finds out, or the teenager or the adult finds out, you know, there's a shock and trauma of their life because they were essentially denied a natural right, which is a natural right to one mother. Okay, one biological mother. So that's built into a 3-parent embryo. You'll see that sooner or later, the inventors of this are probably going to get the Nobel Prize because of advancing humanity and giving healthy kids to mitochondrial disease women. Okay, so it's all framed in context of a justice issue. Of course, no one is considering the injustice to the embryo, right? But that's the reality, and there it is. Okay, let's move to another one. Chimeras. It doesn't just stop with humans. The technology is there. Remember, this is all what is known as the technological imperative, all right? Technological imperative is that anything that can be done will be done sooner or later. That's the technological imperative. It's a philosophical argument. Anything that can be done will be done sooner or later. Of course, it's right down the line of utilitarian ethics, because utilitarian is, as long as the end is good, whatever means, as long as it's legal, Whatever means is ethical because the end justifies the means. Of course, here I am a woman who has mitochondrial disease. Why should I pass that on to my children when I can now have normal children with 3-parent embryo? You know? And so you see the technological imperative. Anything that can be done will be done sooner or later. There's no question of the may. Should it be done? Should it be done, right? Okay, so along those lines, why just stay with the human? Why not human and non-human? I mean, where's the difference, right? We're all mammals anyway. We're all animals. So lest you think that this is total science fiction and the professor has gone over the edge now, in 2016, just before those presidential elections precisely, because of the possibility of the change in administration or the continuation of administration, whichever way you want to spin it. But this was the end of the tenure of the 8 years of W, okay? Now, there was a moratorium which W put into place for not using federal funds for human embryonic stem cell research, okay? So he was trying to protect the human embryo, consistent with the party line of being pro-life and all that. We know that we're in a bipartisan system and the social issues are divided, one for and the other against. This is no secret. This is total public knowledge. So W had put into place a moratorium on using federal funds for human embryonic stem cell research and every other research that would involve the manipulation or the killing of human embryos. And so federal funds mean specifically 2 big agencies, NIH and NSF, all right? NIH, National Institutes of Health and Prevention in Bethesda, Maryland. They're the ones who are involved in human research, medical research. NSF, National Science Foundation, also now it's in Arlington, just moved out of DC. They're involved in funding non-human research in the United States, all right? Now, to the tune of billions of dollars. Last time I looked, which was about a year ago, the NIH budget was about $13 billion annual, $13 billion. That's 10 to the 9, okay? NSF, these are the 2 largest research funding agencies in the world, NIH and NSF. But it's at the federal level. In other words, the feds have nothing to say with regards to state funds and with regards to private foundations. So there's still money there, but of course, NIH and NSF are what we call the deep pockets for research in the United States and throughout the world. So this is a proposal from NIH a couple of months before the elections in 2016, that this moratorium be lifted specifically for creating, manufacturing chimeras in the lab between a human and a non-human animal, between a human and a non-human mammal, all right? So they're looking at 2 models. You remember the Genome Project? The Genome Project started in 1990, a very ambitious project to decipher the genetic code, the human genetic code, in other words, the human genome, right? The 3 billion base pairs that make up the human genome. That started in 1990, and it was supposed to be a 10-year effort back then because the sequencers were primitive and all that. According to today's, computers used to be like the size of a refrigerator and a lot of advancement in technology. Anyway, I remember because I was in graduate school, well, in the 2000s is when I started thinking about the genetics doctorate. At any rate, it was 99.99% complete in 2000, right? But it was not 100% complete. 0.001% of 3 billion base pairs is still quite a few million base pairs. And so They got an extension, I think, for about a year or two, and Francis Collins was the one who took it to completion. I think to this day, he's still the head of NIH. But Francis Collins was involved in the Human Genome Project back then, and the project at some point ran aground.
I read it down like a few days ago.
Sorry?
Yeah.
Jorge? Question? I think he's...
No, no question, sorry.
Okay, okay, I think your microphone is unmuted. Anyway, yeah, so at one point what happened there with Human Genome Project is that two camps developed. So these are sequencers that were going on throughout the whole world. There were labs, not only US, Canada, Australia, Israel, Europe, of course, India, many, many labs throughout the whole world that had sequencers. They were sequencing the human genome like crazy, right? To piece it together. And then at some point, two camps developed. One camp wanted to maintain the sequence public and for free, online, right? With the free access mentality. And that's actually how the internet was created back in the, I think it was in the 80s or maybe 70s by CERN, the European accelerator, which is Switzerland and France, anyway. And another camp wanted to keep it private and have it available at a price, okay? A modest price, very modest price, but you could, it's a little bit like what Microsoft did, you know, that patented whatever technology for using Office, for example, or using the OS, the operating system. And so you just charge 1 cent, but you have millions of people using this system, so you get millions of dollars out of it, right? Anyway, those two camps developed and two different mentalities. And finally, thanks be to God, the public access, free public access camp won. And Francis Collins was very instrumental in doing that and taking it to completion. So finally, we have now the genome available. But even the genome originally, you know how many people were sequenced? How many people's DNA cells were sequenced to put together a human genome to represent what were maybe 7 billion people back then, or 6.5 billion people? It was 100. Yeah, it was 100, which is a magical number in statistics. You know, there's really no magic about 100. It's just a convenient number to do a percentage. But 100 people is what contributed to the human genome back then. And that's in there for 20 years now. At any rate, back to NIH. Wanted to relax this moratorium. And the goal here, what is the background? So the goal is, oh yes, that's why I mentioned the human genome, because now there is a brain project going on. I don't know if you heard about the Human Brain Project. Just like the Human Genome Project was in the 90s, okay? In the 2000s, one of the ambitious goals for the third millennium in medicine and science was to learn as much as possible about the human brain, from the atomic level, molecular level, all the way to the thought process and to the philosophical and theological level, if you will. Everything about the human brain that can be known possibly, all right? And it's called the Brain Project. And there are many labs throughout the world that are involved in this Brain Project. But one of the things that is needed in a Brain Project is what? A brain on which to experiment and on which to test and so forth. And so with living human beings like you and I, we're limited in the research that can be done on a brain, especially since the rise of IRBs, institutional review boards, and bioethical committees, and consent, and free and informed consent, and all this. So therefore, we're now more and more limited on the research that can be done on a human brain. And even if we want to go on a model that is similar to ours, but not quite, the closest one, of course, would be the chimpanzee brain, which is very tiny compared to ours. We have about 3 times the volume of a chimpanzee brain, but that would be the closest in the animal kingdom. And again, you can think of all kinds of reasons why we should not be excessively experimenting on chimpanzee brains. let alone the human brain. And so NIH and many other labs in the world who are in this brain project are eager to develop a brain in the lab, and that is a human brain, okay, without a human being attached to the brain so that there's no consent required. And so they figure out, well, why not? We can do it already. We can do it. Why not develop a human brain in an animal and generate this chimeric model, animal model, all right? And they were targeting the pig, the pig. Because the pig has apparently the same size of a human heart and has more or less the bulk of a human in I guess, some other characteristics that are similar, and they're fairly easy to raise because you feed them garbage and so forth. So let's generate a substantially human brain in a pig. How's that for a concept? Number one. Number two, they still wanted to explore further all the issues about human reproduction. And so it was also convenient to develop a chimeric model where we would have an ample access, not to human sperm because sperm is very cheap, human eggs, without having to go through the whole hyperovulation process and the risks to the mother of the woman and having to pay her and the consent and all that and all the concerns about women's rights and the exploitation of the poor. So how about we grow ovaries, human ovaries in a mammal, in a pig? And we have huge volumes of now human eggs available for research coming from a non-human. Okay, so those are the two desires. Those are the two models, chimeric models that were desired, but minor detail, there was a moratorium on doing that with federal funds in the United States. And so NIH, being federal funds, in other words, being our tax money, put out a request, put out a survey to see if the American people would like to relax that moratorium, okay? Because after all, the money comes from the American people, comes from our taxes. So they put out this survey asking the question, should NIH funds be involved in the production of a chimeric human, non-human chimera for the purpose of research only? In other words, they were only going to grow these embryos to a certain stage and then destroy them, kill them, because they just wanted to prove the principle that it could be done. Mm-hmm. This is the actual wording, okay? Research in which human pluripotent cells capable of generating any organ, right, are introduced into non-human vertebrate embryos up through end of gastrulation. Then the asterisk. NIH will not fund research introducing human pluripotent cells into non-human primate embryos, monkeys, through the blastocyst stage per stem cell guidelines, the ones that were in place, prohibition, but committee would consider introduction of human cells into non-human primate gastrula stage embryos. In other words, bypass the blastocyst, just go right to the gastrula. What do we have a gastrulation? A gastrulation, we have the basic body formation, right? There's the elongation of the ICM into a head and tail region. And there's also the bilateral symmetry establishment at the gastrulation. So what does that mean? Oh, here's the head region. So we can target the head region for the human ICMs, pluripotent cells, to see if we get that monkey brain to develop some human characteristics, because it will be a humanized monkey brain with ICMs introduced, pluripotent cells. You get it? That's what's going on organically, biologically, gastrula. So forget about the blastocyst. At the end of the day, it's the blastocyst that is the moratorium, okay, the prohibition, so we don't have to worry about that. Then research in which human cells are introduced into post-gastrulation non-human mammals, exclude rodents. So here the mouse is out because the mouse is too simple a model, not human enough, where there could be substantial contribution to animal brain or substantial functional modification to animal brain. Substantial, not accidental, but substantial. In other words, to humanize an animal brain, to humanize a mammal brain, okay? And exclude the rodent because the rodent is not human enough. So who is next on the line? Well, of course, the primate. All right. So they sent the survey out actually to selected individuals in the American population, individuals who would be essentially utilitarian and in favor of this. But at one point, some member of the Knights of Columbus picked up the survey and sounded the alert and the red flag and sent it to Supreme headquarters of the Knights of Columbus. And then from there, it was disseminated to all the councils of the Knights of Columbus throughout the United States. And I got the email. And so I continued. Then I spent several days sending it to my own email baseline database about the survey. And so what happens is that the NIH got an overwhelming response of no's, because then the general public did realize, where are we going? What are we doing? You want to use my tactics to create a human brain in a non-human? No way. Okay, so the NIH, the thing backfired and they got an overwhelming no from the general population. And so it was shut down. But then what happened? In November, there was a change of administration. So forget it, by executive order, this was opened up and the floodwaters opened up with the change of administration. So that's the, you see that these bioethical issues have a social impact, they have an economic impact, and they have a political impact. There's no way we can avoid that, okay? Because we human beings are social, economic, and political. So at any rate, in California, at the Salk Institute, because they have also millions of dollars of funding and the NIH funds were not available at that time, they decided to proceed with the experiment actually. And what they did was they generated the blastocyst here. Actually, they generated the embryo, but not implanted. And then they sent that chimeric embryo to Spain, where there are plenty of pig farms, because jamón, bejugo, and all this is very famous in Spain. They eat a lot of pork. And so the pig was raised in Spain. And this is the actual pig with the humanized cells. Here is the procedure taking place. Here is the blastocyst, as you can see. This is a pig blastocyst, all right? And these are the human blastomeres that are pluripotent, in other words, human ICM disaggregated that is being inserted. Here's the target region where already a hole has been perforated through the membrane. And that microscopic needle is going to release these human blastomeres inside the pig blastocyst with the hope that some of these blastomeres, human blastomeres, will attach to the ICM, but it's gone shot because back then, we're talking 2016, CRISPR was still not available, okay? And so it's shotgun blastomere, human blastomeres into a pig blastocyst to see where they implant. And they were tagged, of course, these were tagged with some kind of bioluminescence. Here's the pig embryo. And this is the actual article from Cell, which again is one of the most prestigious journals, scientific journals in the world, with the breakthrough event. Again, the chimeric embryo was generated at Salk in California. and then sent to Spain for the gestation itself. And then they dissected that embryo to see where the cells had gone. Okay, so these are the chimeras. Of course, from a utilitarian perspective, is justifiable because the end, imagine if we're able to grow a human brain in a non-human animal, we can do all kinds of experimentation there. If we're able to grow reproductive organs, human reproductive organs in a non-human animal, again, we have excess of those gametes available. So from utilitarian, it's good. From principle bioethics, of course, not good. Intrinsic evil, because it's a huge offense to the human dignity to do this. All right, the consciousness, the very soul of the human person may reside in the brain. And what are we doing when we're putting the human consciousness in an animal? There was a movie actually about this many years ago. Back then it was science fiction. It was called The Island of Dr. Moreau. I don't know if you ever saw it. Maybe in the 80s, or I don't know when it was. Let me see. And then there was a sequel done that I think was just called The Island a few years ago. Yeah, there it is. This was the original, right, of creating these monsters, 1990s. It was an island. These mad scientists, right, started doing this type of chimeric thing. And so you had these monsters running around in the wild at night and freaking people out. Somebody gets lost in the island and they're hearing these grunts around and so forth. So that was the original in the 1990s. Then I think there was a sequel to it that I think it was just called The Island, which had even more science fiction to it. Just curious. Yeah, there we go. This gets even more sci-fi. All right, so you can check it out. Lots of fun, but now can be done in the lab. Back then, fiction.
I heard yesterday in the news that they were experimenting with CRISPR in order to preserve animals that are in extinction.
Yes.
And they're actually doing it right now. I'm trying to do the same with prehistoric animals.
Yes, yes, exactly. So CRISPR is a fascinating technique. Again, Nobel Prize is there. It's amazing how essentially now scientists can take any segment of DNA from any species and insert it into the genome of any other species, but targeted. Instead of being shotgun like this, right, where we don't know whether cells are going to integrate into the body of the host, now targeting a specific region. So we can take, let's say, human brain genes and insert them into the pig brain gene sequence, all right, where that brain is going to be formed. So they will express in situ, they will express in particular organs. For decades now, there has been a project to try to reconstruct the mammoth. But that one was very primitive by today's standards. What's happened, so we need not just fossil, but we need actual tissue. We need some DNA, some fragment of DNA, right? And if it's intact, of course, much better. And since these mammoths have been found frozen in Siberia, then we actually have tissue. We don't have just fossilized, mineralized bones and heart tissue, but we actually have soft tissue with DNA in it. And also Neanderthals, there have been discovery of some Neanderthal tissue, bone marrow inside the bone, okay? And Neanderthal is very close to us genetically, phylogenetically. And so they tried to, the primitive way of doing that was to do essentially a nuclear swab on an elephant egg, on an elephant ovum. Enucleate the elephant ovum, which is the closest relative to the mammoth, and do a nuclear swap, a somatic cell nuclear transfer from the mammoth nucleus. It hasn't worked. And they tried and tried and tried, and it's pretty expensive to do all that manipulation. It hasn't worked. But maybe now with CRISPR, they can really target, okay? But the key factor, the essential element, of course, is going to be the actual DNA, the actual genome from the extinct species, right? Some recent extincts, the passenger pigeon, and I don't know if there's anything left from a dodo around that was from Australia and some species. You know, recent extinctions may still be around some tissue, but going forward, preserving endangered species. They're still living, but are, you know, very few and become fewer. Preserving that DNA, there's a very active area of research going on there. So Jurassic Park may be around the corner in some sort, maybe not 70 million years ago, but maybe something closer. Who knows? Who knows? And that idea is not too far-fetched. Remember, the idea of Jurassic Park was to take DNA from insects, right? From mosquitoes, from biting insects, because reptiles, so our RBCs are enucleated. Mature RBCs, they ditch their nucleus. That's why they last so little in the body. They die out soon. But reptilian RBCs are nucleated, all right? And so at first I was confused. I said, how can they use RBCs if RBCs are nucleated? Yeah, but only in the mammals. In reptiles, the RBCs are nucleated, all right? Mature RBCs. So those somehow survive the decaying process of apoptosis or whatever self-digestion is going on, then potentially, I mean, it's hypothetically, it may have some merit to obtain genomes from biting insects way back then. Anyway, the next one is growing human organs in animal models. Now, this one, maybe we're not growing a whole human being and we're not growing a brain. So how about growing a pancreas, a human pancreas for people who are diabetic? How about growing a human pancreas from islets, you know, in a mammal that could regenerate the pancreas of a healthy individual to be transplanted into a person with diabetes. So as long as we're not dealing with the brain or with reproductive organs, that's possible. And this was actually done in the 1990s. There is the vacante mouse. However, there's a twist here, okay? So these mice, first of all, these are called nude mice, and they're nude not because they don't have any hair, or they have like a little fuzz actually, and typically they're albino, but they are stripped of the immune system. That's why they're called nude, because they're stripped of the immune system, and therefore they have to be kept in very sterile environments and so on and so forth, right? But they're used for research, especially in transplantation research, so that they don't reject the foreign body, the foreign object. And what's the foreign object in this little vacante mouse? The foreign object is an ear lobe, an ear flap, right? Not just a lobe, it's an ear flap. In other words, essentially is cartilage, all right? It's an ear flap, but there's no hearing here. The mouse is not hearing through this ear, okay? Because it's non-functional. There's nothing behind that ear flap. There's no middle ear, there's no inner ear, there are no tympanic membranes, there are no the little ossicles, the little bones, there's no nerve, it's just a flap, okay? So to call it an ear is a little stretch of the imagination. It's an ear flap basically to see if it can be used for reconstruction of the ear flap. Someone who has an accident, gets their ear scraped off or something, it can be reconstructed, all right? So that's the idea there. But we're still a long ways from generating an actual functional organ, human organ, in an animal model. But this is kind of the first step in proof of principle by using nude models that don't have an immune system to reject the precursor of the organ. Maybe it can work, all right? This went back in the 90s, as you can see. So some people are attempting that. Okay. It's 11:15. Why don't we take a little break before we go into the next two? We only have two more to cover, basically, which is going to be prenatal diagnosis. Oh yeah, this got inverted. Give me a minute. No. Yeah. Anyway, yes, we're up to here. And we still have prenatal diagnosis and fetal medicine. All right, we can take a little break here. We may end a little early today. Okay. So how about we take 15, we'll come back at 11 and wrap it up. Any questions, comments, anything? Okay, let me pause and we'll be back in about 15 minutes, okay? 11 a.m. Okay, everyone, welcome back. Here we go for the final stretch. And we want to cover prenatal diagnosis and fetal medicine and surgery. So, prenatal diagnosis has been around for some time. In fact, this is what motivated my first dissertation in Rome when I was doing my first doctorate in moral theology. And because back then, I'm talking about the early 90s, the technique had just come on to medicine as a standard practice. And the question was, well, with prenatal diagnosis, one can determine a number of illnesses and disease on the human embryo or the human fetus, and then what to do with that information, right? So since it was new, then I decided to do my dissertation on that because for, as you know, for a PhD, in contrast with a master's, one needs to do some original work and advance the field, as they say. And the ERDs had just come out. The ERDs are the ethical and religious directives, directives. Back then, I think it was like the 5th edition. They're up to the 6th or 7th. Let's see, 2018. This is here from the United States Conference of Catholic Bishops to USCCB. Back then was, I believe, was the 5th edition. And prenatal diagnosis was a first there and was positive in the sense that as long as the results were not being used for aborting the child, aborting the baby, then prenatal diagnosis may be ethical, may be licit, because it gives information for the parents, okay, and for the expectant mother. So basically, what do we have? This is a way of determining how healthy the fetus is, or the baby. But the pregnancy is already going on. So it's not pre-implantation as can be done with in vitro. This is an in vivo natural pregnancy that is advancing. And so first we have to look at some of the methods and then the bioethical evaluation of that. So basically the methods are of two kinds, either they're invasive or non-invasive or minimally invasive, right? Invasive carries some risk to the fetus or the mother, and non-invasive are supposed to carry no risk or negligible risks. So the two non-invasive are ultrasound and maternal blood serum, alpha-fetoprotein. This AFP is in the mother's blood, but it comes from the fetus. There are proteins, there are factors that come from the fetus into the mother's circulation through the placenta, right? And so a high level of AFP may indicate some abnormality with the child, and therefore that could trigger a procedure to do an invasive procedure to see what is going on with the child, all right? Ultrasound is very standard now, essentially it's non-invasive, and there's essentially no risk to the mother or to the child. And it's an imaging technique, so that ultrasound is very effective and powerful today. To where they can visualize the human embryo, the human fetus. It's amazing with 3D and 4D. 4D was actually moving. It's just incredible how well they can visualize how sophisticated that ultrasound has become. Okay, these show actual images. And they can even zero in on certain structures, on heart structure, the formation of the spinal cord, for example. So I can start detecting some issues with the baby in utero. Non-invasive. Of the invasive, there are essentially two kinds, amniocentesis or chorionic villi sampling, CVS. And they carry different levels of risk. CVS has more risk than amnio because amniocentesis is essentially inserting a needle through the woman's abdomen into the amniotic fluid and pulling out some fluid, some liquid from the amnion, which is mostly water. But what do we have in the amnion, in the amniotic fluid? We have, go back to ultrasound. In the amniotic fluid, we have fetal cells floating around, mostly from the skin, maybe shedding cells. And so in the fluid there are cells of the fetus that are floating around that can be sent to the lab for examination, for analysis, chromosomal analysis, et cetera, and see if there are any abnormalities, okay? That's amniocentesis. It has some level of risk because it's either transabdominal or transvaginal. They can do another one that is transvaginal. Either way, because it's compromising a little bit the amniotic fluid and there's a puncture on the amnion, the amniotic membrane, it could trigger a miscarriage. It can trigger contractions and the woman could be a little higher risk of miscarrying the baby, okay? Also, this has to be guided by ultrasound, of course, because you have a fairly large bore needle going in there, and if that needle touches the fetus, could do some substantial damage, right? And so it's a delicate technique, it carries some risk. I think back then, when I was studying this goes back about 20 years, 25 years, it was about a 5% increased risk of the woman miscarrying or the mother miscarrying. So there had to be some very significant reason for doing the amnio. CVS, chorionic virilis sampling, is a little bit more invasive. This is actually plucking out some cells from the chorion. So the amnion and the chorion are different membranes of the embryo, and they surround, but the chorion is near the placenta. And because of that, these are, the villi are hair-like structures, okay? And so they pluck out some of these hair-like structures to, again, to get actual fetal cells and to test fetal cells in the lab. But they're actually invading tissue, they're fooling around a little bit with the placenta, and so it's dangerous. And it has a higher risk of miscarriage. I think back then was about 10% risk of miscarriage. The technique may have been perfected at this time, not up to date on it, frankly, but there's some risk to the child and the mother, but especially to the fetus for miscarriage. Okay, so it has to be justified for some very, very grave reason. I'm going to get into what that grave reason could be in a minute, but here is the actual directive. Is directive number 50 from the Ethical and Religious Directives, ERDs, where it gives the okay for a proportionate reason. Okay, so there's no disproportionate risk. This is a negative, does not subject them to disproportionate risk. So the diagnosis provides information to guide some preventative care for the mother, for pre or postnatal care. Now this is before fetal medicine or fetal surgery was available. So this is projecting into the future, and that's why my dissertation was done on this. Essentially the topic was the human fetus as a medical patient. All right, so the fetus as a medical patient, which was totally hypothetical back then. It was not being done, but you could see it looming on the horizon that it could be coming, all right, that the fetus would be a medical patient. And one of my points in the dissertation there was that, well, if the fetus, the human fetus is a patient, how can we how can we not afford a legal status to that patient, right? That patient has certain rights, natural rights, and therefore should be protected under the law as a patient. At any rate, what we have here from one perspective, the fetus is an ideal patient from several perspectives when you think about it. For example, it doesn't require hospitalization because the mother is a hospital and her womb is a hospital room, right? The hospital bed is being fed by the mother and cared for in a sterile environment. So to provide that to a normal patient in a hospital is really very expensive and technologically intense, and yet the womb provides all that. Plus, the fetus himself or herself is very tender tissue that is plastic, is dynamic, it can be regenerated fast and quickly. And so there are all kinds of advantages there. Also, there are no external insults or co-insults to that organism yet because he or she is not born, so they have not been exposed to a substantial amount of UV, for example, or toxic chemicals, anything like that, that we are normally exposed to once we're outside the womb, okay? And so from a number of perspectives, biological and clinical, fetus could be an ideal patient. Of course, the medicine and the surgery has to be very well calibrated because we're talking about a very tiny human being, and particularly surgery. The instruments have to be very large. well, enlargement, which can be done. You know, I don't know if you noticed, some doctors today, even dentists, sometimes they use lenses in their eyeglasses. They use like little microscopes also, they can enlarge the image that they're working on several fold, 5-fold, 10-fold, you know, it's huge. So that same technology can be used. In fact, that technology is used for microsurgery that is done, repair of a number of parts of the body that are very tiny. So from that perspective, the fetus can be an ideal patient. So the prenatal diagnosis as a diagnostic tool may be very useful in determining what protocol to follow to heal that damage on the embryo or the fetus, mostly the fetus, before that baby is born, okay? So we get into this last area where we can look at the human fetus as a medical patient, and then we have to do a cost-benefit analysis. Again, I was writing my dissertation in the early 90s, and by the second half of the 90s, already there were foundations being established to deal with maternal fetal medicine, and even a journal of maternal fetal medicine that is out now. It's a scientific journal, it's a medical journal, okay? So some examples are here. Typically, shunts and stunts and tiny surgery can be done. Well, again, medicine and surgery. So medicine, fetal medicine involves medication. Giving medication to the mother that will go through the placenta and target the baby, target the fetus for some kind of healing, all right? Or could be injected directly into the fetus or the amniotic fluid for the fetus to absorb. That would be fetal medicine as such. But surgery is another level of sophistication. And here are some examples. For example, little balloons that are put into the lungs when the lungs are not developing normally to encourage the pressure on the lungs for normal development and so forth. These are little tiny shunts that are put in, or balloons that are inserted with very small instruments, ultrasound guided, if it's done in vivo in utero, all right? Or in an extreme case, the uterus is actually pulled out live. The uterus is disconnected, right, from the tubes, or maybe they leave the tubes intact, I don't know, but disconnected from the vagina and pulled out and then opened up, an incision, surgical incision, and the fetus is worked on and then sewed back together and reattached to the mother. It is amazing. It is amazing, and there are the photos. There's one very famous photo of this first fetal surgery that was done like this, extracting the uterus and working on the baby. And when the surgeon was getting ready to sew up the wound, right, the womb wound, the fetus stretched out his hand and grabbed the surgeon's pinky, okay? And grabbed the surgeon's pinky. The original was a pinky. Grabbed it like this, like, I don't know, people interpreted thank you or something, but he actually grabbed the surgeon's pinky like that. Everyone was amazed, everybody taking photographs of that. And then it went viral because it went on the internet. And since then, it has become kind of popular, or kind of a trademark of these fetal surgeons to put a finger on the fetus hand so that the fetus would naturally curl around it. You know, it's like kind of a trademark for these fetal surgeons to do that. But the original one was like that, was done spontaneously. He just reached out and grabbed the pinky. Anyway, it's a further level of humanizing that patient, right, that fetus. There are other techniques, a little more sophisticated. For example, when you have twins and they're sharing part of the placenta, there's vascularization there that one twin tends to develop bigger and faster than the other twin because actually his or her capillaries are cannibalizing on the placenta of the other twin. And so the other twin doesn't grow as fast, it's actually being malnourished. So they can now go in there and cauterize these vascularities, these blood cells, so that each fetus placenta stays on their own side and feed only their own baby and not cannibalize the other nutrients of the other baby, okay? So that's another type of creative surgery that can be done. The meningoceles, these are all different bubbles or fluid accumulation that occur when the spinal cord doesn't develop normally, when that, the formation of the spinal cord, remember it comes from exoderm, right? It comes from the skin pouring into the neural groove. It's a process that's called invagination, and that exoderm is pouring in and generating the neural cord, and that's going to become the whole nervous system, the central nervous system, the spinal cord and the brain with cephalization at the forehead. But when that process is not done homogeneously, there can be some little pockets that develop, and those are known as meningoceles that accumulate fluid and they bulge out, and then they can stunt the growth of the further development of the parts of that region. If it occurs on the lower part of the spinal cord, for example, it can interfere with nerves that go into the legs and can cause paralysis on the child. If it occurs more in the cervical region, it could cause paralysis on the upper arms, on the upper limbs, again, because impinging these bubbles of fluid will press on the nerves that are trying to develop there. Sometimes these pockets develop inside in the brain, like hydrocephaly, and then fluid in the brain will cause the brain to develop abnormally, all right? So now they can be drained. They can be drained with little shunts and tubes so that the pockets can be taken care of in utero, and then the embryo, the fetus, can continue normal development, okay? So they're getting very creative with all these kinds of surgeries and medicine going on. And it's a whole field now. Maternal fetal medicine is a whole field of specialization with their journals and everything else. So at least something positive on the intervention of the human fetus and the human embryo, recognizing him or her as a patient, all right? And therefore afforded the dignity that she deserves. So I wanted to end up with something positive, and you can see now how foresight this document was with the ethical and religious directives back in the early 90s to foresee that this was coming around the bend, and therefore, as long as prenatal diagnosis is not used for an abortion, then it could be positive, all right? Even in the case where there is no cure for whatever illness, for example, Down syndrome, Dr. Jerome Lejeune, who discovered trisomy 21, to his dying day, he wanted to cure that. He wanted to find a genetic way of trying to block that extra chromosome 21 in utero, so he would take care of Down syndrome babies, all right? He called them his little patients, and he was very pro-life and totally destroyed by the fact that his discovery has led to the killing of so many human beings in utero by the mere fact that they are Down syndrome children, okay? But unfortunately, in that sense, Down syndrome has not been able to be cured. But if at some point in the future, that extra chromosome can be isolated, right, then that would be because they would just afford the normal expression of two chromosome number 21 instead of the extra load of the third chromosome. Okay, anyway, so here's some advantage for prenatal diagnosis. When I was doing my research back then, and this of course is dated, back then, the vast majority of PND came back negative, meaning that there was no issue with the child, okay? So we have to understand diagnosis. When a diagnosis is negative, it's a good thing. It means that whatever they were testing for did not show up, all right? So whatever illness or condition they're testing for, if we get a negative result, that's good. So a negative result is positive in that sense. It means that we have a healthy development, or at least for the thing that they're testing for. When we get a positive result, then a positive result is bad news. In other words, it means that whatever they were testing for came back positive. The child is carrying that illness, that disease, that condition, all right? So a negative result is positive, a positive result is negative in medicine, and that's how it's worked out. Well, when I was doing my research again back in the early 90s, the vast majority of PND came back negative, thanks be to God, over 95% of it, meaning that the pregnancy was developing normally. Why is that? Especially when it's tested at the fetal stage, all right, beyond the first 2 months into the second or third trimester, most of those babies were developing normally. Why? If, on the other hand, we have globally, the human species has globally about a 50% pregnancy loss, natural pregnancy loss. In other words, that half of human pregnancies don't make it to term naturally. I'm not talking about a procured abortion. What are those? Spontaneous abortions or miscarriage, early miscarriage, right? It's just a technicality before or after the 8 weeks, the 2 months. But the vast majority of pregnancy losses occur really in the first 2 weeks, around the event of implantation. And again, many of those carry some kind of chromosomal abnormality. So remember, it's a type of natural selection. The body, the woman's uterus, naturally rejects that embryo that is carrying some kind of abnormality. There's no guilt involved. It's just a natural process. And I've mentioned typically because it occurs, many of them occur in those first 2 weeks of fertilization. Sometimes the woman doesn't even know she's pregnant. She may just think she's late in her period, but she's actually having an early pregnancy loss, all right, of a little embryo that had some kind of genetic disorder. So it's natural selection. And like I said, globally, it's about 50%. Depending on who you read, again, it's kind of hard to quantify very accurately because many of those early pregnancy losses, they're not counted as pregnancies, they're just counted as maybe late periods or something like that, you see? At any rate, with such a high pregnancy loss, prenatal diagnosis could be something positive when it's, as long as it's not used for aborting. But the 2 stats that I wanted to share with you was that the vast majority of prenatal diagnosis came back negative, which is a good thing for the mom and for the couple, right? They're carrying a normal pregnancy because the abnormals have already been discarded. But of those who get a positive diagnosis, well, about 95% of those abort, okay? And that's the negative side. When there is a positive diagnosis of something that is wrong with the child, the vast majority of women end up terminating that pregnancy, killing that child through abortion, okay? Now, what do we have? PND can test for so many things. When you start at the minimally invasive or non-invasive first and move from there, it could be something as simple as missing a few fingers or missing a finger or missing a hand. Doesn't show up on the ultrasound, all right? That's certainly compatible with life, right? People can live without a finger, without a hand, without an arm, without a leg. But of course, the couples, the intuition is they want the perfect child. This child is gonna suffer. Let's put them out of their misery before they suffer, please. So that's one extreme, you know, something that is as innocuous as missing a finger, for example, which can be detected later in just through ultrasound, or something as drastic as missing a brain, right? Microcephaly, for example, or anencephalic, which is some babies make it through birth, anencephalic, but they're missing a brain, so they're not going to last long, sometimes a few minutes, a few hours, a few days, a few weeks, but eventually that baby's going to die, all right? And so most people, sadly, couples, of course, most couples don't really have a substantial biological education or even theological or religious education to keep those pregnancies and to keep those children until God takes them. So they end up jumping the gun and killing the child instead of allowing the child to die. If it's not compatible with life, then that child is going to die sooner or later. But even if they're born just for a few minutes or a few hours, at least it may give the opportunity to baptize that child, right? Because we don't baptize in utero, but once they're born, they can be baptized. So anyway, these are the heart-wrenching questions when a couple undergoes PND. And therefore, the big question to begin with is, why do PND? You know, why do PND at all? Because if the result comes back positive, then what are you going to do with that results? There's a temptation, let's get rid of this pregnancy, right? So the couple, what I'm trying to say by all this is that really a couple who undergoes PND, or a mother, a pregnant mom who is going to go through prenatal diagnosis, she better be very solid on her values and principles before she gets the result, just in case she gets a positive result. What's she going to do with that? Okay, and so it really is challenging, to the point that I actually know some couples who the doctor has suggested, well, they see something on the sonogram, that's the first level. So that sounds the alarm. Let's do the second layer, which would be a biopsy, either through amnio or CVS. And the couple is pro-life. So say the couple says to the doctor, no, doctor, we're going to stop here because if the child has any abnormality, we're keeping the child anyway. So why put the child through a risk of even just a slight higher percentage of a miscarriage if we're not going to do anything about it anyway? So forget the amnio, forget the CVS, we'll continue with the pregnancy and we'll take whatever God sent. All right, and so that's a pro-life attitude, right? At any rate, some couples do want to be ready because there are support systems, there are support groups for Down syndrome families. For example, I have a child in my family, she's a second cousin, Eliza, and she's the life of the family. She's a teenager now in high school, fully mainstream in regular classrooms. Of course, she has a tutor that is with her all the time, but she's mainstream in the classrooms. And her mom decided, my cousin decided, we're not going to do prenatal diagnosis for her because we're keeping her no matter what. And she actually is a member of a team that is a support group for families who have children with Down syndrome. Dade County, Broward County, you know, there are these local support groups. So they party together, the kids get together, and so their kids look normal to each other because they all have it, and things like that, okay? So a lot of support groups out there in different conditions that is healthy and promotes life and promotes the dignity and the sanctity of human life, like we say, at every stage of development, from conception to natural death. And natural death is what we're going to be looking at in 2 weeks. So I think this could be a good place to stop. Any questions, comments? All right, so we've covered the full gamut here, more or less. The issues keep coming. Of course, technologies was driving a lot of this. Always keep in mind that technological imperative because it filters into society very easily. And there's a link between technology and economy, right? Technology stimulates the economy, and between economy and politics. And so at some point, technology is driving economy, and the economy is going to drive some kind of policy. And then that's why Aristotle talks about homo politicus, in the sense that the person who lives in the polis, in the city, in the civilization. we should argue these points before we dictate law that then is the law of the land and this respects humanity one way or another, all right? There are a lot of issues at the beginning of human life that point toward a disrespect toward that human embryo who is anthropologically, ontologically as human as you and I. We have sometimes we're challenged with that because they don't look like us, but they look exactly how they're supposed to look at that stage of development. So a zygote looks exactly like a zygote should look like at that stage of development. Doesn't make him or her any less human. Okay, we'll encounter these issues at the end of life with increasing technology also, and how it's moving from technology into ideology and from ideology into policy. But typically what is happening there at the end of human life, it's moving at a state level. Instead of being federal, uniform law, like for example, abortion being the law of the land at the federal level, all right? Some of these camps have wisened up, and what they do is they introduce it at the state level. Like abortion was before Roe v. Wade in 72 up to 73. Some states had legal abortion and other states did not have legal abortion, okay? So that's what some of the groups are doing now with end-of-life issues, going at the state level because those are easier battles to fight, okay? All right, well, thank you once again. I'm closing here. And also a renewed invitation for coming to do some restoration work in the forest next Saturday, starting at 9 a.m. I'll be there from 9 until about 2 p.m. You can bring your breakfast if you want. You can bring your lunch. Don't bring your dinner because I won't be there for dinner. Okay, but come dressed accordingly. And then whatever other tools we use, clippers and loppers and everything else, I'll provide that. All right. Thanks again, and have a good rest of the week.
Thank you. Have a good weekend.
Thank you. God bless. Bye-bye.
Bye.